Good day everyone, welcome to the course on biological data analysis and visualization with R. In the last class we started working on the ChIP-seq data analysis. We started with data exploration. So, we looked into two data sets and we have started with one of the data sets where we are working with BAM files, and we will be following that up with all the specific steps including binning and peak calling. So, let us continue that, right. So, we will go into the peak calling part. So, the topics that we will be covering in this class, the first would be the read binning part once we have loaded the data, and then the peak calling part. Okay, so, just to remind you again of the flow chart, right. So, this is something we have seen. We have to start with the read mapping data, the BAM files, and convert them to bit files. We have seen how we can do that, and we have to do the binning, then peak calling and visualization. Okay. And again, this is something we have very briefly discussed, how we can use BET tools to convert read mapping data from BAM file to bit file, and what are the operations that we can actually run on the bit file. Okay. So, we will pick it up here from in this class. So, we will first look into this data set that we have generated, the bit files that we have modified, and then we will actually load the data using this import bit function, and we will use the fragment extension, which is the R package ChIP-seq. Okay. Then we will follow that up with the peak calling part. So, we will have to call peaks in the treatment sample compared to the input or the control. Okay. So, here we have this H3K methylation, right, specific methylation that we are working with versus the input control. So, we will use peak calling based on the input control, and then that will give us the binding regions, likely binding regions of the protein of interest or the specific histone modification mark. For peak calling, we will be using this tool called MAX3. So, again I will show this tool before we actually go into the peak calling part, and this is a Python based tool. Again, this we can run through the Linux command line. Okay. So, this is the command that we are going to use, the MAX3 call peak -t treatment, the treatment file that we have to give, and the -c control file, the input file that you have to give. The file format is in bed format, the -g is the genome size, right. So, here we are using, so you can specifically mention what should be the exact genome size, or we can mention, right, which genome we are using. So, here we are using Drosophila melanogaster, so we just mention DM. If you are using human genome, we can say HS. If you are using mouse genome, MM. So, this notation, short notations we can use instead of trying to mention the exact genome size. And then there are some options we have given, for example, minus no model. I will show you why this is minus no model, because this is kind of recommended by the tool. Then we have -n out, the output file base name. Okay. This is something we will go and do. Okay. So, once we are done with this peak calling, in the subsequent class we will actually visualize the data using ensemble genome annotation. We generate data tracks and we will plot these data tracks with counts and peaks. Okay. For ensemble genome annotation, we use this package Biomart. Okay. So, this is something we have introduced in one of our earlier classes, where we discussed this bioconductor package Biomart, right. How do you actually find this Biomart, the locations of genes, and how do you actually load these genomes using this Biomart package. So, this is something we will do, and then we will define gene locations, etc., promoter regions, etc., and then we will use the R package GViz for visualization. Okay. This is kind of the very brief outlook of what we are going to do in this class as well as the next class. Okay. So, let us start now with the hands on. Let us go back to the bed files, and then we will first do the let us load the data, then we will do the binning and the extension and peak calling, right. So, that is what we are going to do in this class. Okay. So, let us move to the command line, right, and as I showed you, right, we have now generated these chromosome 3r.bed files, right. These are the bed files that we have generated in the last class, where we have chosen chromosome 3r. Okay. This is where this gene, target gene, is located. Of course, this will change depending on your gene of interest. Okay. And you can of course generate multiple such files depending on if you have multiple genes of interest that are present in different locations in the genome. All right. So, I just want to show you, right, how these files look like. And as you have seen, we have chosen only this chromosome 3R, right, not the other chromosomes, because this is the chromosome which we are interested in, where our gene of interest is located. Okay. Okay, so let's load these bed files now in R. So let's move on to R and start working on this data set. Okay, so here is R. Okay, and as I discussed in the last class, the first step is to get this genome annotation, right, DM6, this genome version DM6 that we have to load here. This package I have already installed from Bioconductor. So this should load here in R, and then what we will do is we will load this, right, genome, we will store this in genome, right, this genome variable from which we can extract information. Okay, so this package is now loaded, and with some information. Okay, so this is what we are loading now in this genome variable, and we can see what we have here. Okay, so we cannot see head, but what you can see is the length. Okay, sequence length. So what we'll get if we run this head sequence length is the size of the chromosomes. Okay, so here are the size of the chromosomes, as you can see in Drosophila. Chromosome 2L, this is the size. Chromosome 2R size, chromosome 3L, 3R, 4X. Okay, so these are the sizes of the chromosomes. Okay, all right. So we have loaded the genome version now DM6. What we need is the sequence info of this genome. Okay, and this is the sequence information. Okay, maybe head doesn't work because. Okay, so we have these 1870 sequences from DM6 genome. Okay, all right. So this is what we have stored here. Okay, the next one is we need the feature map for fly genome. Okay, and for that we will use the Biomart package. Okay, so this is something we have seen before, how we can load this, and at the same time I will also load the GVisp package, which is for visualization part, which will be used later on. Okay, so all these packages are now installed from Bioconductor R, and once we have loaded this Biomart package, we can use this function useMart, and this is something you are familiar with. We have used this before. Okay, what we have is useMart, the Biomart, right, ensemble mart ensemble, and then the data set. We need the Drosophila data, right, Drosophila melanogaster gene ensemble, and this is the link, this is the archive link. Okay, so this will load. I wanted to show you these archives where you can find them, right. How do I know this is the, we have to use May 2024 archive. Okay, so if you go to ensemble archives, right, here it is. Okay, here we have ensemble archives, and you have the versions that are stored here. Okay, and May 2024 or September 2024, these are the latest ones. But why do you want to see this? Because for some archives we have to use the earlier versions, right. So for some data analysis, if you want a specific version, then you have to use some of the earlier version. For example, if you see this version, October 2014 version, so it's an old version. And if I, let's say, choose the fruit fly genome here, okay, you can see this version is an earlier version, BDGP 5. Okay, the one that we are using and the data set was using is the DM6 or BDGP 6 version. Okay, so that's why if you have a data set where an earlier version of this genome builds were used, then you have to use those archives. Okay, all right. So let's now go back to this R. Okay, there is some issue, it seems, and maybe something to do with the connection. Let's see. Okay, so once we get this connection done, then we'll look into this feature map. We'll load this feature map from the package, right. So we'll generate this feature map and we'll store this ensemble gene ID from there. Okay, and then what we'll do is we'll define this location of this gene, right, METL3 location from biomarker gene region track. Okay, and we'll specify chromosome 3R, genome version TM6, the start position, right. So we have the start position here. We'll give the start position slightly before the actual location of the genome of the gene, and the end position slightly after the end of the gene. Okay, and the biomarker that will be used, then some of this name should be ChIP-seq, the colors, etc. This is something we'll use and the feature map, right, this feature F map that we'll define, that we'll use for visualization later on. Okay, so this gene region track we can define now, and we'll use this for visualization later on. Okay, so let's see if we, okay, so this is fine now, this is loaded now, we use this biomarker, and we'll now define this feature map. Okay, first is the GFIS, okay, feature map, and the F map symbol, okay, is ensemble gene ID. Okay, so you can see this once we have this bmart loaded, what we can see is the list attributes bmart. So this is the command, right, 1:10, right, or 15. So first 15 attributes we are going to see. Okay, and here we have these attributes, the name of these attributes. For example, the first one is the ensemble gene ID, transcript ID, and so on, right. Start position, end position, chromosome name, all these things are here. Okay, and transcript start, transcript end, transcription start site. So all these things are mentioned here. Okay, and here is the explanation for this. So transcription start site is the TSS. Okay, all right. So we have now also got the feature map. Let's see if we have feature map. Right here we have these different parts of this feature map. We have the gene ID, transcript ID, 5' UTR start, 5' UTR end, 3' UTR start, 3' UTR end. Right, all these things that are given. Okay, that are loaded here. Okay, so now what we do is we'll just store this location of this gene, right, METL3 location, as we are interested in this gene. You can define for other genes as well, right, whatever you like. And then this is the command, right. This gene is located on chromosome 3R. The genome version is TM6. The start position, end position, the biomart that we have just loaded, this bmart, and then size 4. These are for visualization that will be useful. Okay. Okay, and the feature map is also added here. Okay, so these gene locations, etc., 5', 3' at end, etc., all these things are there. Okay, and the data that we are going to use is this use data set. Okay, and we are loading this. What you will see is this UTRs is this will be used later on when we actually identify the promoter regions. Okay, so when you want to identify these promoter regions, we'll use these UTRs, right, in our analysis. Okay, and okay, so this is once we have this, so we want these features of the UTRs for chromosome 3R. This is where our gene is located, and then we'll get this again similarly stored in this UTRs variable, so that we can access this later on, right, when you want to look at the promoter regions, etc. Okay, so let's see when it loads, and then once this is done, we'll start loading the data itself. As you will notice, we'll need a few more packages, right. For example, genomic ranges, R-track layer, I ranges, etc. These are some of the packages that we need. Okay, so this is now loaded, and let's now run this here again. Okay, so we are defining these UTRs. These are the attributes that will be stored here: start position, end position, chromosome name, strand, transcription start site, etc. Filters will be chromosome name. The values should be this chromosome 3R only for chromosome 3R, and the mart should be this data set, right, that we are using. So, this is the D-melanogaster gene ensemble. Okay. All these commands will change depending on the data set that you are working with. Okay. So, we are now done with loading the genome build and genome annotation part. What we are going to use now, actually will load the actual data. Okay. And before we do that, we need these 3 packages: genomic ranges, R-track layer, I-ranges. Again, you can explore in Bioconductor what these packages actually do and why they are useful. So, just simply search and you will see these packages here. Okay. And so, representation and manipulation of genomic intervals, right. So, because we are working with these genomic intervals, genomic regions, we need this genomic ranges package, and similarly you will see why we need this I-ranges package, and the other one is the R-track layer. Okay. So, maybe something to do with okay, yeah. So, yeah, again, so this is an R interface for genome annotation file and the UCSC genome browser. Okay. So, this is what we are using because we want to use this whatever data analysis data results we get, we want to put that with respect to the genome annotation, right. We would like to know where these peaks are, etc. Okay. So, we have loaded these 3 packages now, and let us load the data, and this is the function control import dot bed, right. So, first this is the control data or the input data, then the next one is the replicate 1, and then we have the replicate 2. Okay. So, that is why we are storing them in 3 variables. One is control, the second one is replicate 1 R1, then we have the R2 files. Okay. So, let us load this. Okay. So, okay, maybe we are not in the right directory. So, this is why we are getting this error, right. So, we are in this data analysis in R. So, we have to go into the right directory, and we have okay, right, and we are now in the right directory where we have the data. Okay. And let us run this command. Okay. So, it seems there is some okay, because within that we again these files are not here. So, that is again the problem. Let us see, analysis from BAM, this is where the data set is. Okay. And let us run this now. Yeah. So, this is we were not in the right directory. So, that is why I could not load them, but now once we are in the right directory, we are loading the data. We have the input file, then we have the replicate 1 and replicate 2 file. Again, because these files are quite big, it will take a bit of time to load the data sets. Okay. So, once we load the data sets, then we can have a look at this first, right, the data set and the overview of the data set. Followed by that, what we are going to do is something called the extend. Okay. So, this extend and trim function. So, what it will do, it actually take the read and it will estimate the fragment lengths, right. So, because we are working with the data, right, we are as I said is we are working with data where reads map, right, surrounding the actual binding site. So, these reads, using these reads, if we have single end data, we usually estimate the fragment length. Okay. So, this is the something which we will do using this ChIP-seq package in R. Okay. So, we are loading the data. Once the loading is done, we have to first perform this step, and then we have to do the binning part. Okay. So, this is a step by step process, and let's load the data first, and we will first look into the data set. Okay. And then we will do this. Okay. So, in the meantime, as we are loading, so let us look at the ChIP-seq package. Okay. So, this is the Bioconductor ChIP-seq package where we have this data, and you can find this, right. You have the HTML, you have the PDF, you can look into the specific analysis, etc. and one of the functions that we are going to use is this estimate mean fragment length. Okay. Estimate so here what this function does, right? So this is described here. Estimate summaries of the distribution of fragment lengths in a short read experiment. Okay, and this is designed for ChIP-seq experiments, right? And this is also a note of caution that this might not work well in data without peaks. Okay, so this is the function that we are going to use, and what we need is these are the parameters, and this is again the description of these parameters are given. Okay, which method should be used and what are the different parameters, right, that we have to specify. Okay, and then you can also have some examples. For example, you have this data estimate CS test method equals to coverage. So there are different ways this mean fragment length can be estimated. So we'll just leave it with the default option in our analysis. Okay, we'll just leave it with the default option in our analysis. We'll not giving any command, but please feel free to explore these parameters and see how these results actually change, right? So this is something that will also help you understand what these parameters mean. Okay. All right. So once this is done, let's first look at the data, and then we'll use the library ChIP-seq, and then we'll do the binning later. Okay. All right, which will be followed by the peak calling, and once we have the peak calls, we can then go into visualization. Okay. All right, so let's first look into the data. We're still okay. So this is done now. As I said, because these are really big data sets, they will take a bit of time. Okay, so let's first look at this. Okay, this is how the data looks like, right? When you load this data, we have added this chromosome term already, right? So CHR3R, because as we have seen in the ensemble genome builds that we have loaded already, they also have this CHR3R, CHR2L, etc. So then we can kind of compare this annotation here with the actual data. So here are the regions, right, where we have and the strand information and the score value that are given. Okay, so this is what we have loaded, and we have checked this. We can also check the length. Okay, so this length function is giving us the total length of this file. Okay, all right. So this is what we have. So we have a bit more data in the HTK36ME3R2. Okay. All right. So we have now loaded the data, and we have seen, okay, the loading is fine, data looks fine. The next step is to actually run this extend and trim function, right, where we'll extend the fragments, right. So we'll first estimate the fragment lengths, and then we'll extend the lengths, right, of these reads. We'll extend these reads. When I say trim, means this will also trim the reads if, for example, the fragment length is, let's say, 200 base pairs of 500 base pairs, but the end actually crosses the end of the chromosomes. So in that case, it should trim, right? It should trim to the up to the end of the chromosome. Okay. So this is something that's what we will do by this extend trim function. Okay. The median length is defined by the estimate mean fragment length of the sample for the sample. Okay, and then we have to calculate this median read length, and then we'll have to resize these extended reads. Again, this is a function that is there in the ChIP-seq library, ChIP-seq package. Let's see where this resize function is. It should be here. Okay, there is a resize function somewhere. Anyway, yeah. So here is the resize function, as you can see, right? And we'll use this resize function, and then we get the extended reads, and then the final trimming is done, which will be now returned. As I have shown you, this is an R function, right? This is a function that we write, customized function, our own function, and then for each of these samples, we'll call this function to do the same operation, right? Extend and trim. Okay, and this is what we are going to do here. Okay. Okay, so again it's taking a bit of time, the extend trim function. Okay, median length is 14, it's saying. Okay, so then we have for the next one, okay, for R1 is also 14, for R2 let's see. Okay, read length is 12. Okay, so we have now done this extend and trim part, right? The next step is to do the binning. Okay, and for this binning we need this DMSEQ file, right? This is the DMSEQ variable where we have actually stored the sequence information. Okay, so for sequence in DMSEQ, what we have is this chromosome 2L, chromosome 2R, 3L, 3R. What are the lengths of this each of these chromosomes? Okay, then you have each circular pulse, the genome build version DM6 is given. There is some also some fragments, these are undefined, right? These IDs are given. Okay, so we need this, and the idea is when you say binning is that we divide the genome into these specific beans of certain size. Okay, and in this case our bean size is 500. Okay, of course you can define different bean sizes, right? It doesn't always have to be 500, you can have 100, 200 and so on. So what we are doing here in this part, in this command, is we are creating these beans of size 500 base pairs using this function tile genome, the DMSEQ chromosome 3R, only chromosome 3R, right? The width of this tile should be bean size, right, 500 bases, and then cut last tiles, right, in chromosome. So if it is crosses the chromosome length, then just cut that, right, trim that. Okay, this is what we are doing. Okay, and then we can check these beans, right, how they look like. And as you can see, for chromosome 3R we have created these beans, right. So here we have the 1 to 500, 501 to 1000, right. These are the beans or the tiles that have been created now. Okay, so what we will do now, simply we are now going to kind of count, right, how many reads are in these beans. Okay, so this is what we are going to do. We are going to give the bean samples, right. So for each sample we will call this specific function, and it will actually score these beans, right. So how many reads actually are overlapping with these locations. Okay, so maybe there are 3 reads that are mapping with this 1 to 500 region, overlapping with this 1 to 500 region. There may be 5 reads that are overlapping with 1001 to 1500 region. Okay, so this is what we are going to do here, and this is again a function that actually is scoring these beans, and this is doing this by count overlaps beans sample. Okay, and then it will return these beans. Okay, this is a function that we again apply to all of our samples, right, and this function can take 2 variables. One is the sample name and the beans that have been created. So beans have been created, right, these are also stored in beans variable. So for each of them we are sending this sample name, right, the control and the beans information, and for all 3 we have sent this now. Okay, and what we can do is we can now look into these beans, right. For example, we can again now okay, let me see the file names. Okay, so this is not in capital, sorry. We want to see this one, right, where we have created the beans and we have scored according to beans. So you can see this now, the score has changed, right. So we have added this score, and these scores are counting the overlaps or number of reads that have mapped to these beans in chromosome 3R. Okay, so we have this for the full chromosome now. Okay, so this is the binning part, and then we can do something, right. We can look into these binning profiles, right, and we can look into the specific location, okay, where our gene is located, and we can simply plot this and look at the this core distribution within that genomic region, right, where this MET-L3 gene is present. Okay. So, for that what we are getting is the bin location, right, where we will have this. This is, if you remember, slightly before the actual location of the gene, and we are actually dividing by the bin size, because this is where we will find this gene, right. So, bin log from and by 2 we are dividing by bin size. So, in that which bin we will have this location stored, and this is then we can use this plot function bin location from to bin location 2, and then the score values we are plotting, right. Score is simply looking at how many reads, right, per bin we have, right. That is what we have in the Y level, and this X level, sorry, this would be chromosome 3R. Okay. So, we are looking at chromosome 3R. So, this is chromosome 3R. Okay. So, what we are looking is the X value is bin log from to bin log 2, right. So, we have location from to location 2 the bins, and then the score values, right, for this bin from one bin to another bin. Okay. And then the data point values are given. Okay. So, as you can see, right, this is the location of chromosome 3R, and you can see this read count. So, here it is close to 0 in this region, and then here we have some counts, right, read counts per bin. We will see this in much nicer way later on, but at least we have some reads that are present around our gene location. This is something we can see here, and for chromosome 3R as well we see the same thing. For the control we can also run very similar, and we can see what should be whether we have these reads and so on. Okay. So, this is something you can do. The last thing we will do is we will just export this now into bed graph format, right. After we have done this binning, we will store this information into bed graph files. So, we can actually export this, right, this bin information after the binning, the scores we have into these files. Okay. And this is the command export, right, this variable control bins 500. This is where we have done the binning already, the file name where it should be stored, and the format should be the bed graph format. Okay. And this is what we have done, and for the control as well as for the 2 samples. Okay. So, what we have done so far is we have loaded the data, we have now done the binning, and we have also seen some of this read coverage in this specific location of the gene. Okay. We can now do this even more, right. We can now start looking at this with the genome annotation, and this is something what we will do, right. We will visualize, right, where our gene actually is, how this information looks like how this read count looks like around this region, and so on, in a much nicer way using this library GVs, and then we will also do the peak calling using MAC3. Okay. So, that's what we will cover in the next class. So, let us now go back to the presentation. Just to summarize, this is something what we are going to do. We have done the binning, and after the binning we have to do the peak calling, right, and this is what the will kind of complete the full pipeline, where we will get the BAM file, we will start with a BAM file, and then we will do the peak calling, and we can then visualize the data along with the genome annotation. So, this is something we will do in the next class. Thank you.